Good morning or afternoon to you, and thank you for joining today's webinar. We are Ilse Roodink and Debby Kruijsen of Immunoprecise Antibodies Europe, and today we are going to present an update on IPA's COVID program, in which we highlight how the utilization of IPA's multi-species discovery strategies has led to the identification of clinically suitable multi-antibody cocktail formulations targeting SARS-CoV-2. This webinar is part of the Antibody Society's Antibody Discovery and Development webinar series, which can be accessed through their Learning Center. We are available to answer questions during and after the presentation, so please add any and all questions to the Q&A box in the viewer. Answers will appear in the similar box. An on-demand version of this webcast will be available in a few days, and a link to it will be sent via email to everyone who registered. First, I would like to share our disclaimer, as this presentation will be making forward-looking statements. Immunoprecise Antibodies, IPA, is a multinational contract research organization with strong expertise in the full chain of antibody discovery, development, and lead engineering and manufacturing services to support our clients globally with their needs in therapeutic antibody discovery campaigns against proteins in every target class in a variety of diseases. Our end-to-end capabilities are visualized in this flowchart. After identification of a target, we will conduct a druggability analysis to design an efficient and robust antibody discovery and development program. We will guide the program from target tool generation, such as target expressing cells, soluble target proteins and off-targets, and functional assay setup, the antibody discovery phase, including immunization of different species, identification of the lead antibodies using a diverse range of species agnostic antibody discovery technologies such as B-cell select, hybridoma, and phage display, after which the selected leads will flow into further in-depth lead characterization, optimization, and engineering modules, all the way to manufacturing and preclinical evaluations. In the past 37 years, we have performed over 4,000 antibody discovery programs with a success rate of more than 95% and a high client retention using the multitude of optimized innovative technologies and methodologies at our locations in Europe and North America, and thereby built a great portfolio of clients globally, including 12 of the top 20 pharma companies. This success is a result of significant investments during the last 4 years in research and development in IPA's antibody discovery and engineering platforms, and strategic acquisitions of uProtein Express in 2017 and Modicrest Research in 2018. In 2019, we created Talem Therapeutics. Talem is a wholly owned subsidiary of Immunoprecise Antibodies and is focused on the discovery and development of next-generation, fully human therapeutic antibodies targeting infectious diseases, neurology, immuno-oncology, inflammation, and rare diseases using the proprietary antibody discovery platforms and innovative technologies housed at Immunoprecise Antibodies. As visualized in this pipeline overview, Talem initiated a significant number of programs and accelerates these novel antibody treatments to the clinic through strategic alliances and partnerships. One of Talem's advanced programs is the PolyTope SARS-CoV-2 program. This program was launched in January 2020 and was designed to deliver a therapeutic antibody-based solutions that will provide a safe and long-lasting protection against current and emerging variants of SARS-CoV-2. Although there is an urgent need for vaccines to help transmission of SARS-CoV-2, vaccines do not work for everyone as they rely on the individual launching a robust enough immune response to generate their own protecting antibodies, and most likely do not protect individuals with weakened immune systems, such as people with chronic medical conditions and elderly. In addition, vaccines normally do not provide any clinical benefit to patients already infected. And we believe that therapeutic antibodies can fill this gap and will play a critical role in combating COVID-19, since they can be administered to a broader population of individuals and can be effective when administered pre or post-infection and can protect populations who do not benefit from vaccines. To provide a more durable protection against SARS-CoV-2, our goal was to build an antibody cocktail therapy instead of a monotherapy. This is because single antibody treatments, even potentially neutralizing ones, are vulnerable to being escaped by a single point mutation in the virus. And since viruses mutate rapidly, this is a serious limitation of monotherapy. And to overcome the ongoing threat of an ever-evolving virus, combination therapy is more effective because the use of multiple antibodies can broaden the epitope coverage of the viral protein and reducing the risk of mutagenic escape, and engaging multiple mechanisms of action to increase clearance by more efficient opsonization. And we call this our PolyTope antibody therapy. To select diverse, high-value antibodies for building antibody cocktails, we set up a SARS-CoV-2 tailored program. In this program, we have designed and built different antigen tools and used multiple parallel approaches, knowing that their complementary use would cover any blind spot inherent to each platform as visualized in this flowchart. By implementing early functional analysis, promising candidates can be selected early in the discovery chain and allows a smooth flow of the molecules to preclinical studies. The antibody discovery, workflow, and the results will be discussed in more details in the upcoming slides. As mentioned, multiple different tools were generated, and different recombinant spike proteins, such as the fully assembled spike trimer, S1 and S2 subunits, the receptor binding domain, and N-terminal domain fragments, mutants, and spike protein from related viruses, as well as associated spike trimer of the SARS-CoV-2 virus, were expressed and validated. To support the design of antibody cocktails, assays for comprehensive epitope landscape profiling have been set up, such as specificity screens against a multitude of different current and emergent SARS variants, and epitope binning experiments by ELISA and BLI. In addition, cell-based pseudovirus neutralization assays were integrated to allow early functional screenings in the monoclonal phase of the program, and ACE2 spike protein blocking assays were set up using HTRF. To anticipate on the emerging viral mutants and to have the ability to screen on natively expressed spike protein, cellular assays were designed to quickly identify binding properties of the identified antibodies against emerging viral mutants. Since not one platform is simply the best, multiple antibody sources were utilized in parallel. Diversity was initially created by immunization of different species, such as rabbits and transgenic omni-rats. Parallel sourcing of our pre-existing human library sourced from both healthy donors and autoimmune disease patients, and our naive VHH llama library. To funnel down this polyclonal pre-selection of antibodies to a more defined set of different antibodies, multiple discovery platforms are applied, such as B-cell select, hybridoma, and phage display selection. The major advantage of using different platforms is the sourcing of multiple B-cell repertoires to increase the likelihood of picking up rare clones. Promising monoclonal antibodies were identified by deeply screening the output from all sources using various high-throughput methods guided by our function-first approach. More than 10,000 clones were screened for binding profiles by ELISA. Subsequently, cell-based pseudovirus neutralization assay and ACE2 spike protein blocking assays were performed, and sequence diversity was determined, and relevant clones were recloned into a full-length backbone, produced and purified to ensure high-quality material for epitope landscape profiling and reliable downstream characterizations of combinatory antibody sets. This slide presents the diverse reactivity profiles of individual clones derived from the different antibody sources. Each dot is a single clone, and data derived from source 1, 2, 3, and 5 are plotted for full-length spike ECD reactivity on the x-axis versus RBD reactivity on the y-axis. Reactivity profiles of clones derived from source 4 are plotted with spike S1 reactivity on the x-axis versus RBD on the y-axis. The bottom right plot summarizes the reactivity profiles of the prioritized clones obtained from source 4 and 5 with spike S1 reactivity on the x-axis and RBD reactivity on the y-axis. These plots clearly display selection of monoclonal antibodies with diverse epitope specificity obtained from the multiple antibody sources utilized, and in addition confirms that sourcing multiple antibody repertoires is an advantage. Selected clones were further analyzed in a cell-based pseudovirus neutralization assay. And this plot displays spike ECD reactivity data on the x-axis as a function of pseudovirus-based neutralization on the y-axis. And the top dashed line refers to clones with 50% virus neutralizing capacity, the middle 90%, and the bottom 99% virus neutralization. These high throughput assays for analyzing binding and virus neutralization reveals that the lead antibody candidate pools contain neutralizers with different binding specificities. High throughput ACE2 spike protein blocking assays were performed as well, and to have a better understanding of the mode of action of the antibodies within the candidate pool, virus neutralizing capacity was plotted against ACE2 competition as analyzed by HTRF. These graphs display the selected clones with pseudovirus-based neutralization of more than 50% on the y-axis versus HTRF signal on the x-axis. The vertical line in each graph refers to the HTRF data point of an irrelevant control antibody. All clones with an HTRF signal lower compared to the control antibody, which is left of this vertical line, are ACE2 competitors. Interestingly, not all virus neutralizing clones block ACE2 spike protein interaction, which suggests different modes of action. To support an efficient flow of the identified lead antibody molecules into further characterization and engineering, and to prevent unexpected surprises with respect to developability of our lead molecules, early in silico developability profiling was performed. These analyses provide insights in potential developability issues and might already help narrowing down the list of candidates towards the most promising leads to have the highest chance of success in identifying antibodies for preclinical analysis. These plots present the in silico aggregation score on the x-axis of individual prioritized candidates obtained from source 1 to 3 as a function of hydrophobic moment on the y-axis of the left panel, CDR heavy chain 3 length on the y-axis of the middle panel, or PI on the y-axis of the right panel. Trastuzumab is included as a reference antibody. This data suggests acceptable developability profiles of our lead candidates. Additionally, sequence analysis was performed on the identified lead candidates, and this slide displays the rich diversity obtained with respect to the sequences on the selected functional antibodies and is summarized with the substantial diversity in binding and functional properties of our lead candidates. Next, we performed extensive characterization on this set of clones, which Ilse will present in the next slides.
To accelerate nomination of the final leads, we prioritized our fully human candidates, reformatted to full-length human IgG1 antibodies for epitope landscape profiling and extensive biophysical characterization to assess developability. I would like to focus on epitope landscape profiling first, as these assays underpin the identification of synergistic antibody pairs targeting the spike glycoprotein and our final cocktail formulation. We initially confirmed spike protein specificity by ELISA and BLI. Subsequently, label-free biosensor assays were also used to A. Validate interaction interference with the ACE2 receptor, which is involved in host cell infection, and B. Screen for blockade against a panel of published structural benchmark antibodies. The pie chart on the left shows the merged data from the initial ELISAs and follow-up BLI experiments. Diverse specificities are present within the set of fully human candidates, resulting in a broad epitope coverage across the entire spike protein. The heat map in the upper right panel summarizes a subset of the merged data of the comprehensive epitope binning experiments performed as pairwise antibody or ACE2 competition experiments. This comprehensive epitope binning resulted in the identification of various overlapping and non-overlapping epitope bins across the whole spike protein, which is schematically presented in the Venn diagram in the right bottom. Bin colors and bin IDs presented during this webinar are arbitrarily chosen. To prove that antibodies from non-overlapping epitope bins can coexist on the spike trimer, which is essential for full efficacy of a therapeutic cocktail, we perform two complementary octet-based epitope binning assays. I would like to discuss the data from one of the screening formats, the multi-antibody tandem assay, in more detail here. The presented sensorgrams nicely show a stepwise association of each antibody analyte when applied at saturating levels to the bin X antibody tethered spike. Antibody analytes applied in previously complex forming steps were included in subsequent steps to maintain them at saturating levels. Association analysis of corresponding antibody analytes to naked bin X antibody tethered spike was included for comparison. The shape of the curves obtained for the association of each saturating antibody analyte to naked bin X antibody tethered spike compared with the antibody saturated bin X tethered spikes were identical, demonstrating that each newly associated antibody can bind the spike protein independent of its saturation by other antibodies from non-overlapping bins. These data demonstrate that these four antibodies can coexist at saturated levels on the spike primer, consistent with them targeting non-overlapping epitopes. To exclude any order dependency, we confirmed the simultaneous binding of four different antibodies to the spike protein in a different assay format. Combination therapies that work synergistically and effectively boost protection has previously been described for infectious diseases. As synergistic effects can boost the potency of combination therapies beyond that achieved by the individual components, such effects open the possibility for lowering dosing, which can improve safety. Guided by the epitope landscape, we selected lead candidates from non-overlapping bins across the entire spike protein, not only neutralizers, for in vitro Wuhan-1-based pseudovirus neutralization screening. Many pairwise combinations were rigorously screened using a checkerboard titration method, which resulted in the identification of pairs showing synergy, indifference, and antagonism. We used the pairwise synergies to inform our design of higher order cocktails, which is schematically presented below. Of note, to ensure the quality of the input material, we purified our human antibodies to prevent any confounding effects induced by multimerization. Based on the checkerboard titration data, we nominated several 2, 3, and 4 antibody combinations composed of antibodies belonging to non-overlapping epitope bins for full dose response analysis in pseudovirus-based neutralization assays. Individual components were taken along to facilitate identification of combinations that boost potency, while benchmark antibodies were included for referencing. The titration data of a subset of individual antibodies show that there is a clear variation in neutralizing potency of individual antibodies across, but also within epitope bins. You can nicely see these intra-bin differences by comparing the potency of the green antibodies in the right graphs. The 2 and 4 antibody combinations shown in the middle and right panel respectively clearly show that full neutralization is achieved at a lower total antibody concentration than the corresponding individual antibodies, strongly suggesting synergy. I also want to emphasize that the individual components are present at a lower concentration in the cocktails compared to the single antibody samples. To reduce mutagenic escape risk and engage multiple mechanisms of actions, we aimed at combining as many antibodies as possible that could retain synergistic effects while broadening the total epitope footprint. To this end, we nominated our most potent 4 antibody cocktails and their corresponding individual components for live virus neutralization screening using the D614G variant. As depicted in the color bars on the left, except for one, none of the individual antibodies show neutralizing effects on their own, while upon combination into multi-antibody cocktails, these same antibodies synergistically neutralize authentic virus as evidenced by the black bars on the right. 2 of these 4 antibody cocktails were prioritized for in vivo efficacy evaluation in a hamster challenge model. But before moving forward with presenting the outcome of these preclinical studies, I would shortly like to discuss parallel efforts that have been undertaken to empirically determine developability profiles of the antibodies in our lead pool to minimize liabilities and promote clinical success. As Debbie explained previously, following sequence analysis of hits identified during the discovery phase, in silico analysis was applied to already deprioritized clones with predicted unfavorable developability characteristics like aggregation, hydrophobic patches, and presence of motifs for possible chemical modifications. But also initial production and purification characteristics were taken along as developability liabilities. Where these outcomes facilitated lead candidate selection, empirical evaluation of biophysical properties of lead candidates was performed in parallel to determining the epitope landscape and functionalities to narrow down to a final diversified, well-characterized lead antibody pool. Our in-house workflow to assess developability profiles is summarized on this slide. As you would like to monitor liabilities already at an early stage, our workflow is built on both throughput and parallel efforts, consisting of 3 primary branches: ELISA-based reactivity profiling, as shown in the left arm. The middle branch shows HPLC-based screening methods focusing on retention times and their correlation to developability factors like multimerization and non-specific interactions. And the third arm on the right consists of thermal stability, solubility screenings, and BLI-based self-interaction analysis. Except for solubility analysis, all the methods in this first part of our developability assessment workflow only require a limited sample amount. As we are well equipped to run these screenings in a high throughput way, we could easily assess developability factors of our diversified set of human lead candidates at an early stage. For each analysis, antibodies with a deviating outcome compared to a well-behaving biopharmaceutical antibody were flagged. Antibodies which were flagged multiple times were omitted from the next phase of developability profiling and further functional testing. Antibodies which were not deprioritized during the first phase moved forward to the next phase. While the forced degradation analysis, in which samples are subjected to several stress conditions, including free stalling, different temperatures, and pHs, is currently ongoing for our SARS-CoV-2 program, in general, we evaluate the impact of the various stress factors using HPLC-based methods and mass spec analysis. Based on these profiles, leads are optimized by purposeful design of de-risk variants to minimize developability liabilities. For programs requiring affinity maturation and/or humanization as well, we can nicely apply a full molecular optimization approach addressing these three aspects in one effort. Obviously, developability profiles of any optimized variants will be evaluated in a similar workflow. While stress testing is currently ongoing for the individual components of our multi-membered antibody cocktails as well as for the blend, I would already like to discuss some data from the initial high-throughput developability profiling. For referencing purposes, we included control antibodies with known developability challenges as well as well-behaving clinical stage antibody, referred to as biopharmaceutical antibody. High-throughput assessment of the reactivity profiles of lead candidates toward double-stranded DNA, insulin, and BSA by ELISA revealed that a few clones of the initial panel were flagged. But importantly, as demonstrated in these graphs, none of the individual components of our prioritized cocktails showed binding in this assay, while the polyreactive control antibody shows a dose-dependent reactivity towards all three codings. Next, we examine the potential self-interaction of lead candidates using a high-throughput BLI-based assay format in which the antibody of interest was captured on sensors to subsequently monitor association of the same antibody when applied in solution to the sensor. Although responses, as plotted on the y-axis, are in general low, self-interaction of the individual components is absent or minimal, while a control antibody with known self-interaction showed clear binding signals. This slide focuses on the high-throughput assessment of developability factors using HPLC. Size exclusion chromatography HPLC was performed to determine monomericity of lead candidates. For the full antibody panel, including the individual components of the therapeutic cocktails, over 95% monomericity was observed. To determine colloidal stability, we performed standard monolayer chromatography, HPLC, and observed that over 90% of the full antibody panel showed retention times within the 5% range compared to the biopharmaceutical antibody reference. As indicated with the orange circle, one of the bin B antibodies is flagged as it shows a split peak profile and a longer column retention. Of note, this antibody sample is known to contain a VH glycan PTM, resulting in a heterogeneous sample. Therefore, light optimization of this antibody is ongoing in combination with formulation buffer scanning to improve the developability profile. Clearly, a control antibody with developability issues, captured with the red box, shows significantly longer column retention, strongly indicating an aggregation and precipitation-prone molecule. The antibodies were also analyzed for cross-interaction with human poly-IG, representing naturally occurring serum antibodies using cross-interaction chromatography HPLC. Over 80% of the full antibody panel, including all individual components of our prioritized cocktails, showed retention times within the 10% range compared to the biopharmaceutical antibody reference. Again, A control antibody with developability issues captured with the red box revealed significantly longer column retention. As indicated, the PAC4000 precipitation assay consumes more sample and was therefore performed for a more defined set of lead candidates. Also, while other antibodies were screened at 1 mg per ml, the control antibody with reported developability issues was screened at 0.4 mg per ml due to a lower stock concentration. However, you can still nicely observe that the antibody turbidity plotted on the y-axis developed at a clearly lower PAC4000 concentration for the bad behavior control antibody compared to the others. Except for one of our bin-B leads, all other cocktail components show a similar or better solubility compared to the biopharmaceutical antibody. Based on the PI, we anticipate that buffer scanning, which is currently ongoing, might improve the solubility of the less well-performing bin antibody. Based on the available developability profiles, we applied some light molecular optimization to 2 cocktail components, both belonging to Epitope bin-B, to promote clinical success of our PolyTope SARS-CoV-2 PET therapy. The de-risking focused on eliminating post-translational modification sites, in particular glycosylation sites, in the corresponding variable domains, as these cause sample heterogeneity. For each individual bin-B antibody, a homology model of the antigen binding fragment was generated. The variable domain of the light chain is positioned on the right of each model, with frameworks colored in green and CDRs in blue. On the left of each model, you can see the variable domain of the heavy chain, with frameworks colored in purple and the CDRs in red. For both antibodies, the position of the glycosylation sites are captured with a red circle. For each parental antibody, the cyan-colored amino acids with atom display was nominated for a single amino acid substitution approach. As the spike interaction site is anticipated to be on the top of the model, we expect that replacing selected residues with a gammatically similar amino acid and thereby purposely eliminating the glycosylation site would not impact the spike protein binding capacity of these antibodies. Based on the single amino acid substitution strategy, we designed 1 variant for parental antibody bin-B1 and 2 variants for parental antibody bin-B2. As we felt it would be worthwhile to kill 2 birds with 1 stone, we also made a 3rd variant for the bin-B2 antibody to germline the framework containing the glycan PTM site as much as possible to reduce immunogenicity risks. In this approach, we replaced the cyan-colored loop of the corresponding framework, including the glycan PTM site, with the appropriate germline sequence. The area selected for this loop replacement strategy is captured with a red circle. Following recombinant expression of these lightly optimized variants, we verified whether the binding characteristics were not affected by the introduced mutations. While in vitro virus neutralization screening is ongoing, as evidenced by these graphs, ELISA-based reactivity screening towards WUAN1 spike proteins revealed that the reactivity profiles of the bin-B antibodies was not affected upon light molecular optimization, using either a single amino acid substitution or the loop replacement strategy. The reactivity profiles are plotted per parental antibody. The upper panel shows the bin-B1 antibodies and the lower panel the bin-B2 variants. As demonstrated earlier during this presentation, several of our multi-antibody cocktails are able to potently neutralize authentic virus in vitro. Based on these data, we prioritized 2 of our 4 antibody cocktails for in vivo efficacy evaluation in a hamster challenge model. So let's go back to the functional characterization and discussed the preclinical efficacy data for the two separate hamster challenge studies that we conducted. The upper left visual summarizes the study design in which cocktail 1 was evaluated. Hamsters, 5 ml per cohort, were dosed IP at 40 mg per kg with either antibody cocktail 1 or the individual components 24 hours pre-challenge with SARS-CoV-2 carrying the D614G mutation. Next to this prophylactic approach, efficacy of antibody cocktail 1 was also evaluated in a therapeutic setting by administering the same dose 4 hours post-challenge. This cohort is indicated with TX. As this SARS-CoV-2 model has a transient character and we prioritize viral load as a mean readout, the study endpoint was set at day 4 post-challenge, thereby anticipating that effects with respect to tissue damage reduction might be a bit underestimated, as these effects can be most effectively evaluated 7 days post-challenge. Viral load in throat swab was measured daily by determining replication-competent virus titers. As depicted in the bottom left graph, compared to the mock cohort, animals therapeutically treated with our 4 antibody cocktail showed full replication-competent viral load reduction in throat swabs 3 days after challenge, while only one of the components, when dosed individually, also resulted in significant viral load reduction. The right graph on the bottom nicely shows that, except for one animal, prophylactic and therapeutic treatment of SARS-CoV-2 challenge animals resulted in replication-competent viral titers below the lowest limit of detection in lung tissue isolated at the day 4 endpoint. A similar effect was observed for only one of the cocktail components when administered individually at 40 mg per kg. One of the other cocktail components also demonstrated protection in the animal model when dosed individually, although to a lesser extent. Notably, both these individual antibodies did not reveal any potent neutralizing capacity in a live virus assay using the same SARS-CoV-2 strain, suggesting that cocktail 1 is able to reduce disease in the hamster model via different mechanism of action. To date, no other studies have been published showing in vivo efficacy of therapeutic multi-antibody cocktails targeting SARS-CoV-2. This slide summarizes the main results from the histopathology analysis and nicely shows that despite the early study endpoint, prophylactic and therapeutic treatment of SARS-CoV-2 challenge animals with cocktail 1 reduced inflammation severity of the main air-conducting tissues as represented by the lower bronchitis severity scores captured with a red circle in the bottom left graph. Also, only minimal or mild trachea inflammation was observed in animals post-challenge treated with cocktail 1, clearly indicating that the reduced replication-competent viral titers translate to reduced disease severity in this hamster challenge model. This is further supported by the observation that monotherapy with the highly efficacious cocktail component also resulted in reduced inflammation of the main air-conducting tissues and trachea. In a follow-up study, which exclusively focused on treatment post-challenge, as shown in the upper left visual, we on purpose replaced 2 antibodies by others from the same epitope bin, which appeared slightly more potent in a pseudovirus neutralization assay. Although we reduced the total antibody concentration to 20 mg per kg in this study, you can nicely see in the left graph that therapeutic treatment of SARS-CoV-2 challenge animals with the second 4 antibody cocktail resulted in replication-competent viral titers below the lowest limit of detection in lung tissue isolated at a day 4 endpoint. These data points are captured with a red circle. In addition, to validate the in vitro observed synergistic effects of our therapeutic 4 antibody cocktail in vivo, we included cohorts treated with the individual components at 20, and 5 mg/kg as well. Component D was not evaluated individually, as this antibody already demonstrated no reduction of viral titers after prophylactic administration at 40 mg/kg. As indicated with the orange circles, 2 of the cocktail components, when administered individually at 20 mg/kg, were also highly efficacious in 4 out of 5 animals. Of note, both these antibodies are present at 5 mg/kg in the 4 antibody cocktail, a dosing which only resulted in full clearance of replication-competent virus from lung tissue in 1 of the 10 animals treated with 5 mg/kg antibody A or B. To further support the in vivo synergy, we compared efficacy of antibody A and antibody B when administered individually with a combination therapy of antibody A and B, all dosed at 5 mg/kg total antibody. As plotted in the right graph, only 1 out of 10 animals treated with the immunotherapy showed undetectable viral titers in the lung, marked with a golden circle, while 3 out of 5 animals treated with the antibody A and B combination therapy, captured with a green circle, showed replication-competent viral titers below the lowest limit of detection in lung tissue, isolated at the day 4 endpoint. And we are excited that this study is the first one showing in vivo synergistic effects of multi-antibody cocktails targeting SARS-CoV-2. Coming back now to our initial goal, developing an antibody-based treatment that provides long-lasting protection against emerging variants of SARS-CoV-2. We anticipated that a multi-targeting strategy, our PolyTope approach, would be the best solution to reduce mutagenic escape risk, even if we would face loss of binding capacity of one of the cocktail components. To monitor reactivity profiles of our current cocktail components and remaining antibodies of our lead pool to spike proteins of emerging variants, we use different assay formats and different formats of the spike protein. Reactivity of our leads toward recombinant versions of the spike protein, including stabilized trimer S1 and RBD variants carrying relevant mutations, was analyzed by BLI and standard ELISA. The presented graphs show the full titration curves of the 6 leads of our current 2 antibody cocktails towards plate immobilized full trimer variants. While recombinant proteins might not always represent the full native conformation, which might be in particular true for soluble spike trimer as it contains a stabilizing mutation to prevent furin cleavage, which does take place in vivo, we also verified reactivity of antibodies of our lead pool towards cell-expressed spike trimer variants of concern in a high-throughput way. Using a multiplex cell screening format and a high-throughput flow cytometer, we are able to analyze binding to up to 4 different cell lines in one well. The graphs in the lower panel display the full titration curves of the 6 lead of our 4 antibody cocktails towards different spike protein mutants. In line with the ELISA data, while some individual components are affected by specific mutations, our 2 4 antibody cocktails maintain reactivity towards tested spike protein variants of concern. Of note, the reactivity profiles indeed suggest some conformational differences between soluble and cell-associated spike protein. Overall, diverse reactivity profiles were obtained with quite high epitope bin dependency, as observed for leads in our current cocktails as well, which speak to the importance of high epitope coverage to minimize mutagenic escape risk. Also, relying on high-throughput multiplex cell screenings will enable us to quickly monitor reactivity of leads to future SARS-CoV-2 variants as well. To assess the functional consequence of mutant susceptibility, we tested whether our multi-membered antibody cocktails could retain neutralizing potency in vivo using the original Wuhan-1 and both UK and South African variants pseudotyped VSV particles. Next to the 2 4 antibody cocktails, a hybrid of both cocktails containing 5 antibodies was analyzed as well. The 5-antibody cocktail contains 2 bin-B antibodies to potentially capitalize on subtle differences in their binding reactivities to further reduce mutagenic escape risk. As you can nicely anticipate, despite the fact that binding of some individual components is affected by mutations present in spike protein of the UK and South African variants, all 3 multi-membered antibody cocktails retain potent neutralization in this in vitro pseudovirus assay. Just to summarize, the PolyTope approach for our SARS-CoV-2 program has yielded a diversified panel of antibodies against SARS-CoV-2 as evidenced by the sequence, epitope, and functional diversity. In-depth, early-stage profiling of the epitope landscape facilitated the identification of synergistic pairs and subsequent higher-order cocktail formulation by combining antibodies targeting non-overlapping epitopes across the spike protein. Although therapeutic multi-membered antibody cocktails targeting SARS-CoV-2 have not been previously described, I would like to emphasize that the uniqueness of our higher-order antibody cocktails is also based on the induced synergistic effects, which seems to play an important role in virus neutralization and clearance potency of our cocktails. Apart from unlocking synergistic effects, the broad spike protein epitope coverage of our diversified lead antibody pool allows easy adaptation of the cocktails, thereby strengthening our pandemic preparedness to address current and emerging variants of SARS-CoV-2. Our integrated developability profiling and optimization capabilities were applied in parallel efforts to promote clinical success. We are currently preparing for the final IND-enabling preclinical studies and GMP productions to make our promising therapeutic cocktail available for clinical evaluation as soon as possible. In conclusion, our core strength lies in the breadth of antibody discovery platforms that we offer, which are compatible with several species and targets, combined with our extensive capabilities with respect to early-stage, high-throughput characterizations, including customized functional assays and expertise in lead optimization to minimize developability liabilities and promote clinical success. Our high-quality end-to-end workflow allows us to take any therapeutic antibody program from concept to preclinical batch production of therapeutic leads, as demonstrated today by means of our PolyTope SARS-CoV-2 program. Despite the challenges due to the mutagenic nature of viruses, we believe that rationally designed multi-antibody cocktail therapies will play an important role in combating SARS-CoV-2. Key here is that the included antibodies cover a diversified number of epitopes across the spike protein, and lead nomination is not limited to potently neutralizing antibodies to A, allow identification of synergistic combinations or antibodies with alternative mechanism of action, and B, stimulate Fc-mediated viral clearance by a high antibody declaration level of the spike. In addition, the diverse pool of antibodies that we yielded by our PolyTope approach provides enormous possibilities for plug-and-play cocktails to address future SARS-CoV-2 variants. Therefore, instead of fast-tracking our best antibody as monotherapy, anticipating emerging variants of SARS-CoV-2, we carefully characterize our antibodies for therapeutic cocktail formulation to eventually eliminate the threat of SARS-CoV-2. If you would like to learn more about our SARS-CoV-2 program or IPA's capabilities, please visit our website or contact us via email. Thank you for your attention, and if you have any questions, we are happy to take them in a Q&A session.